Welcome to the lecture series on immunity. So, in the last session we were discussing on innate immunity. So, components of innate immunity we were discussing, in that the phagocytes play a major role in the innate immunity. So, what are phagocytes? These are the cells, example neutrophils, macrophages, monocytes, which will rapidly recruited to the infection site. So, coming to the phagocytosis. So, what is phagocytosis? It is a process of engulfment of microbes and subsequent hosting in phagosome. So, the fusion of lysozyme with phagosome to form the phagolysosome, which will lead to the killing of the microbes. Then coming to the natural killer cells, which is another component of innate immunity. So, natural killer cells are class of lymphocyte, which is essentially we have seen they kill the virus infected cells as well as the tumor cells. Then coming to the mast cells, these are all present in the epithelial lining and they are activated by the microbial products which will bind to the toll like receptors which is present or by the IgE antibody mediated. These mast cells will release cytoplasmic granules which is rich in histamine, prostaglandins and cytokines, and they initiate the inflammation and proteolytic enzymes. So, inflammation is also we have seen in the innate immunity. So, the mast cells, because of the release of the cytoplasmic granules, it will initiate the inflammation and proteolytic enzymes, which will result in the killing of the microbes or the bacteria. So, next one is the dendritic cells. These produce cytokines. Cytokines are nothing but the chemical messengers which will initiate the inflammation. So, if there is any infection, we have seen that inflammation will be there. So, the dendritic cells will produce the cytokines which will initiate the inflammation, and also the transport the antigen from the skin and mucosal sites to the lymph nodes, and they present the antigen to the T cells. So, presentation of antigen to the T cells is a very important phenomena which happens in the body. So, the dendritic cells play a major role in producing the cytokines which will help in the presenting the antigen to the T cells, and also it will serve, these dendritic cells they serve as bridge between innate and acquired immunity. So, next one is the complement pathways. Complements are nothing but some of the serum proteins which will alternate component pathway activated in the response to the bacterial endotoxins. These bacteria will produce endotoxins, and the alternate complement pathway will get activated in response of these endotoxins, and they also lyse the target microbes which is producing the endotoxin. So, this is how they are helpful in the innate immunity. Then they also stimulate the inflammation and they stimulate acquired immunity. So, these are the bridges between acquired immunity and innate immunity. So, next one is the inflammatory response, where you can see when the bacteria, you can see in the picture, the bacteria is entering inside the skin, and you can see that vasodilatation will be taking place. So, that the tissue, you can see the bacteria is entering, and you can see the vasodilatation, and the leakage of the plasma proteins can be seen, which will recruitment of the phagocytes to the site of the inflammation. So, in the picture you can see so many phagocytes, for example, your neutrophils, all these are coming to the site of the inflammation, and engulfment of the microbes and dead materials will be formed because of the inflammatory response, and they will destruct or they will destroy the microbes which is trying to enter inside the body. So, this is inflammatory response, which is another component of the innate immunity. Then the normal resident flora. We know that on our body surface there are so many bacteria are present which are harmless. That means they are called as normal flora. So, this normal resident flora exert several antimicrobial activities and they compete with the pathogens for the nutrition, so that they do not allow the pathogen to enter inside the body or to themselves to stay back in the body. So, because of the competence with the pathogens for the nutrition, and also these normal resident flora will produce antibacterial substances, so that the pathogenic bacteria cannot enter easily inside the body. And the next one is the acute phase proteins. These are synthesized in the liver and the synthesis increases or decreases during acute inflammatory conditions. So, in the any acute inflammatory conditions, that is why we always see for the acute phase proteins, because this is synthesized in increased way in the acute inflammatory condition. So, whenever we having an infection, so it will lead to the increase of acute phase proteins. Acute phase proteins examples are C-reactive protein. So, C-reactive protein, it is an acute phase protein which will rise in acute inflammatory conditions. Most common marker of acute inflammation, that is why when we have any infection or inflammation, we try to check for the C-reactive protein, because it is the most common marker of acute inflammation. The normal level of your C-reactive protein is less than 0.2 milligram per deciliter. So, also it increases several folds with acute inflammatory conditions. Our C-reactive protein can be seen increasing in several folds with acute inflammatory conditions. So, it is detected in the laboratory by using latex agglutination test, where the latex particles are coated with anti-CRP antibodies and CRP will be seen in a given sample. So, we have seen all the components of the innate immunity. So, let us move to the next session on the series called as acquired immunity.